Hey guys, welcome back. We're going to go ahead and do our last video for chapter 19, early evolution. This can be a good deal. So we're talking about protocells last time. We're going to go ahead and take that up to cells and some of the organelles that we see inside those cells today. So the theory for modern genome analysis, basically the idea, of course, is that all living species descended from the same cell. This cell might have lived somewhere in the order of about 4 billion years ago. The cell was probably prokaryotic. Prokaryotes are way simpler than eukaryotes. They don't have a nucleus, they don't have membrane-bound organelles. It's really just DNA, membrane, some ribosomes, and that's about it. We're gonna go with the simplest first. These cells must have been anaerobic because there's no oxygen in early Earth's atmosphere, and that goes back to the whole iron in the rock thing. Um, so they had to live without oxygen, and that's fine. We have prokaryotes alive today that can live in anaerobic environments, so that's all right. There was not much oxygen on Earth, so when we start to try to search for signs of this first life, it's super tough because it's not like a dead dinosaur where it leaves this big skeleton of bones that can become fossilized. When bacteria die, they don't leave a lot behind. If you've tried to look at bacteria in a microscope, depending on how good your microscope is, they can be pretty hard to see when they're right there in front of you alive. So to try to find them in rock is very, very difficult. There aren't very many rocks left that could hold fossils that are from this time frame because it's so long ago. Cells don't have any hard parts to fossilize, and also some of these structures that can be formed just as a rock becoming a rock might resemble a fossil, and so we're very cautious as we look at rocks because we don't want to find some cool pocket in a rock under a microscope and say, hey, that's a fossil of an old cell when it's not really a fossil of an old cell. So we do want to do kind of our due diligence there and make sure that we're sure that the fossils that we're claiming are fossils are truly fossils. So we're pretty certain that we've got some good fossils of filaments. Filaments like maybe from like some algae or something. 3.5 billion years old are these rocks in Western Australia that we're thinking, or we're going to have a pretty good date on them. And here's how we kind of get that idea. And so there's a 3.5 billion year old filament, and it might have been a chain of fossil bacteria from an ancient stromatolite. So what the photo under here, this is the fossil they got of a filament, of algae or something like that, some sort of cell. And this shows these modern day stromatolites in Australia. And basically what you get is these living photosynthetic cells. They trap a little bit of sediment, they die, a new cell grows on top of them, it traps some sediment, it dies. And so these mounds just kind of grow up. And they're thinking that might have been the way this guy was trapped. And they're able to get kind of an idea of what these old cells might have looked like. Here we have these are spheres that we find in sand. And so we're kind of wondering, well, are they just circles in sand or are they bacteria? They might be bacteria, but they might not. So that makes that a little difficult. And so the stromatolite, which I just showed you in the picture, is the mounded structure that forms in shallow sunlit water. It's a mat of bacteria. They trap minerals and sediment. They die. New photosynthetic bacteria grow on top of them, and they increase in size as these new layers cover the old layers. Modern stromatolites do exist today in Australia's Shark Bay. We think that they started growing about 2,000 years ago, and they're standing about 1.5 meters high right now. And so there's lots of different types of bacteria that can carry out photosynthesis. But out of all of those groups, there is only one group, and that's the cyanobacteria, that when they do photosynthesis, they produce oxygen. None of the other bacteria that can do photosynthesis, none of those guys actually make oxygen. Only cyanobacteria do. And so when we look at how much oxygen is present in our current atmosphere, we are forced to give a lot of credit to cyanobacteria. And that's exactly what we do. And so we think that oxygen began to accumulate on Earth somewhere around the order like 2.5 billion years ago. And that's going to be in the air and it's going to be in the sea because oxygen can dissolve in liquid and also, of course, be the gas in the atmosphere. And so what this is going to do is it's going to completely change the sea because when oxygen is around, it is going to be oxidizing metals. And so that includes metals in compounds that are in living cells. And so if you're a cell that can't survive that, that oxygen is going to kill you. And if you're a cell that can use that oxygen as an electron carrier, then you're going to survive. And so this puts a huge selective pressure on the seas, and it's going to increase that atmospheric oxygen. The cyanobacteria will, which will eventually give us an ozone layer. Now, as we get our ozone layer, we get something really cool because now the life that we have supposedly had would have been really deep in the ocean because we need that layer of ocean water just to protect us from the UV rays that's going to mess up our DNA and kill us. When we get enough oxygen and we build up an ozone layer, now the ozone layer way up in the atmosphere is absorbing that UV rays, and life on land becomes a possibility because those UV rays are not going to be destroying you and giving you all these mutations. And so when we try to look for these cells, we try to look for a biomarker. It's just what it sounds like. It's something that marks life that you're interested in. And so this is a substance that occurs only in cells of a specific type of what you're looking for. And so we're looking for when did prokaryotes become eukaryotes? Or when did eukaryotes show up on the scene? And we found a few steroids that exist only in eukaryotic cells and not in prokaryotic cells. And so we can find molecules of those steroids in some ancient rock that we're thinking are about 2.7 billion years old. But as far as the actual cell itself that we're pretty sure we've got a fossil of, we're going to see about 1.8. So there's a little bit of a difference there. Do you go with the steroid existing in the rock, saying, well, the cell must have been there to make the steroid? Or do you go with I'm pretty sure this circle in the rock is the cell, go with the fossil? They're going to give you different numbers. But either way, I guess between friends, maybe we'll call it 2, about 2 billion years. So that's what we'd have to do. And so you guys know that prokaryotic DNA doesn't have a nucleus, so it's just out there in the cytoplasm. Eukaryotic cells, on the other hand, have a double membrane that surrounds their nucleus. It has nuclear pores to allow messenger RNA out. It's very structurally complex. So the eukaryotic cell, their DNA is in the nucleus and it's associated with an endomembrane system, which means we have, rough ER, smooth ER, Golgi apparatus, those kinds of guys who are helping along RNA and protein and lipids and all sorts of stuff they're building in there. And so some of our bacteria have these internal membranes. They're simple, like the membrane just kind of folds into like a pocket, and that's their inner membrane. Some of them do have a little more a thick membrane that actually kind of sits around the DNA, and that can help them. That can help them from biological threats or physical threats. Can help them from UV radiation, things of that nature. So we do see that a little bit in just one or two types of bacteria that we see today. So when it comes to the mitochondria and the chloroplasts that are in our modern day eukaryotes, this is not going to come as a surprise to you because you have done a lot of research and a lot of speaking and listening about the endosymbiosis theory. And so mitochondria and chloroplasts, you know that these guys are like bacteria in their size and their shape. You know that they have their own DNA. You know that they have a singular chromosome. You know that they replicate independently of the cell, that they can move about inside of the cell. And so when we look at all these features, it seems to point to the idea that mitochondria and chloroplasts in the past were bacteria. And the bacteria got into an early cell. And the cell started to rely on the bacteria started to rely on the cell, and we call that the endosymbiosis theory or the endosymbiont hypothesis. Endo's within, symbiosis is having a close relationship, think ecology, mutualism, predation, commensalism, those things from your Gen Bio class. That's endosymbiosis. We're living close together, we're relying on each other inside of one cell. And so that's kind of the idea, and you guys are familiar with that. So here you have a prokaryote, not a lot going on, a little bit of DNA. In comes a little bacterium that's aerobic, and that guy starts to make ATP, and the cell's like, I'm not going to kill you and eat you. Let's just be roommates, and you can make ATP, and I'll keep you safe. And he's like, right on, let's do this deal. And so on it goes here. These guys become mitochondria, which is just awesome. Some of them keep dividing to become animal cells. Others go ahead and do this process again, where little photosynthetic bacteria get into here. And the cell's like, hey, instead of eating you, let's hang out and be roommates. And the photosynthetic bacteria can make food from the sunlight. And the mitochondria can take the food and make ATP. The cell keeps them safe. And they're just one big happy family, pretty much ready to take over the world because they're the best design ever. Cool? So that's endosymbiosis, but you guys probably already knew that. So we look at eukaryotes. We look for cells that are alive today, bacteria, that are most like a chloroplast or that are most like a mitochondria. And what we do find is there are a few types of bacteria that are similar to mitochondria. Oops, sorry, that underline was just crazy. I might be able to find their book in here. Yeah, their name rather for that bacteria in here. I can barely say its name. That's kind of crazy. Let's see, where is it? Here we go. It sounds like rickets. That's why it's so weird. Rickettsia prowazekii, which causes the disease typhus, has a genome that's very similar to that of some mitochondria. And so we think this Rickettsia bacterium, its genome looks a lot like mitochondria, so we think maybe their ancestor was the one who kind of hitched a ride into that first cell and went on to become the modern day mitochondria. The chloroplasts, we think those, of course, I mean, they make oxygen, so they must be derived from cyanobacteria. And then our components of some of our first protists are going to start to show up like a nucleus, like the endomembrane system we just talked about. And then thanks to eating a few of his neighbors without truly eating a few of his neighbors, we get mitochondria and chloroplasts. And once you have all these things, you have the modern day cell. So it takes quite a number of very happy mistakes, and in the correct order, with enough time, maybe we could get a cell. So that's the idea. And so here's some milestones when you look at what are the big events from not life to life. Um, Forming those building blocks, getting those first amino acids, that's a big deal. Getting those first cells, that's a big deal. These are huge steps to make. Diverging domains, thanks to archaea, bacteria, things that are photosynthesizing, things plants versus animals, that kind of stuff, is all going to start from really simple things like whether or not you have a chloroplast. Photosynthesis, having oxygen in our atmosphere, just completely changing everything. Aerobic respiration evolving in mitochondria. It's a huge, very, very complicated process that has to evolve. The endomembrane system needs to get here. The nucleus needs to get here. We need to go ahead and go through endosymbiosis twice in the correct order to get mitochondria and chloroplasts. And then after that, we can get to fungi, animals, plants, you, me, dogs, cats, birds, whales, all of it. Cool? So here you go, 3.8 billion years ago. Some cells keep on being bacteria, some cells become archaea, some cells become what's going to eventually be the eukaryote. Some of these cells here are going to get that mitochondria and become an animal cell. Some cells will get a chloroplast and become a plant cell. Some of them will just continue just cruising on being marine bacteria. They're not going to go any further on that evolutionary chain. They're still alive today. They're still in the ocean. They're still doing what they were doing 1.4 billion years ago. They're still doing that in our oceans. Others change. Some stay the same. Cool? And so there's modern life. That's how we got there. So this little guy here, He's going to give rise to all the fungi, all the animals, protists, everything. So, a couple questions at the end of the chapter here. Evolutionary biologists believe that life on Earth first evolved in the absence of oxygen. Where did that come from? Hopefully, I've said it a few times. You can quickly say cyanobacteria. Duh, we've been over that. And then the tougher one. Can you think of any environment on Earth within which life cannot exist? Hmm. I don't know. Cannot and does not. Maybe two different questions. Let's go with cannot. Okay. All right. So chew that over. That's the end of the chapter. I hope you enjoyed it as much as I did. For me, it raises more questions than it answers, but good questions lead to more questions. So let me know what your questions are, and we'll just kind of work through this together. Thanks for your time.